Holler if I'm too loud or too quiet. There's a nice lead-in by Damien on using genetics for helping to identify a lot of these things. I'm going to kind of give you a broader background on genetics and kind of give some examples as a lead-in to kind of the workshop that we'll be doing later this afternoon. I want to stress that DNA work, genetics, is not a scary topic. Often people think of it as this really high-tech, intensive lab, type of work, but it's something we could all do. We did a workshop in Ecuador in 2010 where we had all the participants sit down at their tables and actually do the extractions, do the identifications in the lab. So it's something that you can pick up in a very short period of time, just like looking at the fin guide that Damien just put out. So it's an easy technique, and I'll try to stress that over and over again through this talk. So in a lot of cases, we have the whole bodies. We have these great guides that we can go through, as Jimmy talked about earlier. We can identify species fairly easily. This becomes a bigger problem, as we know, when we get fins or other pieces. There's problems if you're looking at meat. Meat is definitely a difficult thing to identify species from. But one of the key things is, as Damien said, we don't need to go through and identify all of these thousands and thousands of fins. We can use the keys to help narrow down what we're looking at. So we can go through and find a subset of samples that we suspect might be CITES species. We can go through and spot check a variety of the ones that we exclude just to make sure that we're not missing individuals. This can be particularly important for some of the smaller fins, the non-primary fins. We can also use, as Damien was talking about, we can use the genetics to verify the visual IDs, but this is also a good thing to use in routine just to make sure that everyone's doing well. We can test their ability to visually identify these samples. So DNA, there's two types of DNA that we generally work with. There's the nuclear DNA. There's a single copy inside of each cell. This is the DNA. You get one half of your DNA from your mother. You get one half of your DNA from your father. We also have mitochondrial DNA. So mitochondria are these little cells, or little things inside of the cell. And what's neat about these is they actually have their own genome. So where we have one copy of nuclear DNA, we have multiple copies of mitochondrial DNA. And these also differ in a couple other ways. Nuclear DNA comes from both parents. Mitochondrial DNA tends to come only from the mother. So this has its own caveats in that if you have hybridization, you can be led to misidentifications by using only mitochondrial DNA. The size of the genome is dramatically different. The nuclear DNA, you're talking of hundreds of millions to trillions of bases in length. The mitochondrial DNA, you're around 16,000 bases in length. Mitochondrial DNA has a bunch of different genes that evolve at different rates, and this has been traditionally used for a variety of studies and still being used a lot for population studies. This gene here, the cytochrome oxidase subunit 1 gene, is the gene that's being preferred for a lot of the barcoding work that I will discuss in a little bit. So there's a million ways to preserve DNA. We know that it works fine on dried shark fins. It's not always the best, highest quality of DNA. If we're looking for preserving samples for more in-depth studies, we often freeze samples or preserve in absolute ethanol to get the water out, make the sample be in its best quality state for long-term storage. But there's a variety of other methods out there, either saturated salt solutions or even packing fins in dry salt. The key is that for preserving DNA in samples, you want to get water out. Water is the enemy. So drying these fins, having nice dried fins, tends to work pretty well as long as they're not kept in a moist environment where they rot. And all of these have their caveats where ethanol and alcohol in general is flammable. It's problems for shipping. Frozen samples requires a big infrastructure for maintaining your freezers. We've all had freezers fail and you lose a lot of samples. And then a lot of these other methods aren't great if you don't keep the samples in a dry environment. Typically, to get DNA out of the samples, we do a variety of techniques. It usually involves tissue digestion, where you dissolve the sample either by use of enzymes or by use of chemicals. You dissolve it into a slurry and then typically you use some way to clean up the sample, so you're purifying just the DNA, and you use organic extractions as kind of an older method, using a lot of these organic solvents, phenol chloroform, followed by precipitation of DNA with ethanol. There's also a lot of these commercial kits now that are very simple. They provide all the components necessary, and you mix your sample with a buffer, and it binds to a glass membrane matrix, and you use a centrifuge, and it's pretty simple. They call one of the kit DNA Easy because it is easy. But the method I prefer, and I think is great for a lot of the work that people should do on identifying shark pieces, is a method I use called the Keelex boiling method, where you essentially, I jokingly say, you make shark fin soup. You just take a piece of the fin, you throw it in a tube, and you cook it. And the DNA comes out of the cells into solution, and it works pretty well for a lot of downstream processes. In some cases, in a real laboratory, you might care about the quantity and the quality of the DNA, so you would use UV absorbance using a spectrophotometer to assess absorbance at different wavelengths to look at the quantity and the quality. And this is important for some processes, but for the species identification question, it's not really an important thing. So what's made genetics work well for people over the years is a polymerase chain reaction. This is a relatively new method. I mean, it's been around for 20 years or so, but this is what's revolutionized the genetic studies. And it's pretty simple in its nature. You're essentially targeting one piece of DNA. You have unique primers that are specific to a certain location on the DNA, and by using a process I'll go over here in a second, you can make a lot of copies of one small piece of the gene. And this allows you to do sequencing, allows you to run gels on the samples and do a lot of downstream stuff. So in the simplest example, we start off with a double-stranded piece of DNA. You heat the sample, the two strands come apart. You cool it down. We have these unique primers that sit down on specific pieces of the DNA. And then thermal stable DNA polymerase essentially copies. It adds nucleotide bases to the end of the primers and makes these copies. Each time we do this cycle, we double the number of DNA copies. So we go from 1 to 2 to 4 to 8 to 16. And by the time you get to 35 or 40 cycles, where we typically run a PCR, you're talking trillions of copies. So it's a very efficient way to get a high copy number of specific pieces of DNA. Of course, these days when you talk about genomics, or when you talk about genetics, we think genomics. And this is kind of a big step forward. 10 years ago, we were typically doing one gene from species and hundreds of base pairs. Now with genomics, we can do hundreds of millions of base pairs to sequence the whole genome. And this is getting cheaper every day, but this is not something that we would do for our species identification purposes. But these are new tools that as we start to go out, we can find ways to identify new species, ways to better understand how these animals adapt to different climate changes, the speciation drivers. I just thought I'd give an example. This is a fish that we're working on, the California yellowtail. Each one of these little blips here is a gene. So where 10 years ago we'd focus on one little gene, now we can get hundreds, thousands very easily and relatively cheaply. So what do we do when we have all this DNA information? Well, we've got a variety of things historically that people have used it for. We can do phylogenetic analyses. These are looking at relationships between species, looking at possibly discovering new species. Population genetic studies, this is more of a finer scale look at relationships between populations or management units. And we can start to look at questions, looking at paternity analysis. We can look at mating systems. This is important for understanding some basic concepts of genetic diversity. And kind of an interesting thing is that every animal has a unique genetic fingerprint. So we could track individual samples if we wanted to through the trade as a genetic code. We can actually replace a lot of the standard tags that we think about for animals with these, just get a genetic sample, release the animal, and so we can understand a lot about what's going on. And then finally, what we're most interested in is species identification. So we can use this to validate fishery observer calls. This is important. And a lot of fishery management contexts beyond the monitoring for CITES, but also it's important for, as we're talking about, identifying shark fins, and can be used to identify other difficult things. Eggs and larvae and juveniles of a lot of bony fishes is an important thing. So looking at phylogenetic studies, typically we've used, and we still use, a combination of multiple genes from both the mitochondrial and the nuclear DNA. This provides us multiple levels of evolutionary change. We get, from looking at these and comparing these genetic data, we can generate measures of genetic similarity and use these to construct DNA trees and perform statistical testing using maximum parsimony, likelihood, Bayesian methods to identify the strength of the relationships that we find. So I just thought I'd put an example here. This is the relationships among the hammerheads. This is some of the utility that you can use the DNA sequences for. And if you look in more depth, you can start to look and identify little bits and pieces. This is the new species of hammerhead, as we talked about a little bit. And by looking more in depth at a lot of the samples, not just trying to identify what species a fin is, the more we mine and go through and look at the data of these samples, the more new species we're going to find. And I know that we're finding new ones in shark species every time we look, and in pretty much every species. So this is something that's going to be an ongoing issue, that we're going to find new species. We're going to have to identify that. We're going to have to monitor it. I just threw in, this is the new species of hammerhead as described. Things we have to start thinking about as we look more. We're going to discover new species that are in these CITES complexes, or species that are going to be someday CITES listed. Also, the utility of getting some of this genetic data. If we have sequence data, we can start to look at population differences. So here's an example from Sphaerina lewini. We can start to see that there's some geographic partitioning. So not only can you identify species, but you can go about trying to identify where this sample came from. So now we're getting into the species identification. Ideally, every species has a unique DNA sequence. This isn't always the case, depending upon the different marker that you look at. Different genes, some species share sequences. We have to identify what we want to look at, come up with a consensus idea. And this has been worked on with the Barcode of Life initiative, and the decision has mostly been made to look at the cytochrome C oxidase subunit 1 gene in the mitochondria. This is usually called CO1. And to make sure that we know what we're looking at, we have to have reference material from all species. So this is a daunting task, but it's actually been pretty well overcome using museums and collections throughout the world, a lot of collaborations, and then reference databases of all of the sequences so that we can go through and query these databases with the sequences we get and get our species identification. And there's several methods I'll talk about briefly here for doing the identification. The first is DNA sequencing. This is the gold standard. Typically, we get all of the information we need from that, but it's expensive and it's time consuming. So for what we want to do, what stuff that we would do here and what we'll do later is looking at kind of these cheaper, easier, faster methods using PCR-based and gel-based separation. So DNA sequencing, as I said, it's the best method for getting the most information. You can use this for looking for possible cryptic species. You get a lot of characters there for making sure that you have high quality IDs on your animals. You can potentially get population origin information, and you can use a lot of these data for other studies. It's fairly expensive. There's more steps. You need trained technicians. You need a fairly dedicated lab space. The equipment is not it's not trivial. If you have a DNA sequencer, you're talking hundreds of thousands of US dollars. There's companies out there, and there's one, Macrogen in Korea, you can send your DNA samples to, and they'll process it for you and give you a data file back. So there's ways to make this a little bit cheaper. But in reality, it's still a process that takes 2 or 3 days. But if you have the sequence data, the tools are out there to provide your species ID. So this is the Barcode of Life database. You can look it up on the internet, barcodinglife.org. It's a pretty cool initiative trying to get DNA sequences from every species on the planet so that we can identify anything by sequencing one or a few target genes. If you look at things like this is the current state of elasmobranchs. They've got over 16,000 sequences in here from elasmobranchs representing over 800 species. It's a fairly broad coverage, and you have pretty high probability of finding a match with, or multiple matches when you submit a sequence. So just as an example here, I can take some of my sequence data that I got, enter it into the web browser here, press submit, and you come back with a nice little match. These are all 100% matches to sequences from silky shark, and it gives you kind of a probability of placement within the different levels of taxonomy here, saying it's 100% match with silky shark. So it's a fairly easy method. It gives you a lot of good information, and you know, if you have the money and the time, it's the best way to do things. But of course, we don't always have the money or the time, and so there's other ways that we'd like to do things. So RFLPs, or species-specific PCR, these take advantage of the data that we know about the species. We know the unique character states in the DNA that we can use with restriction enzymes that can cut the DNA into specific sizes, or the PCR that will amplify different sizes of fragments for the different species. This is pretty simple. The training, as I said earlier, it just takes a little bit of playing around with the pipetters. If you can follow a recipe to cook, you can probably do this. Laboratory equipment is relatively minimal. You can bring it in your suitcase. People have done this over and over again, going to different places. The lab equipment to do this is relatively cheap. You can get an entire setup for probably under $10,000, and then once you have that, the actual per sample assay cost is relatively cheap. And I put these numbers here. These are much higher than you would expect. You can actually find better deals if you shop when you look at sales. You buy things in larger bulk, so you're actually less than 50 cents per sample, and it starts to get more into the labor time. And you can get your identifications in usually just a few hours. So as I said, we have the species-specific PCR. It takes advantage of differences in the genetic sequence to amplify different size fragments for individual species. In this example, this is from Mahmoud Shivji's lab. This is one of the first shark ID PCRs out there. It control primers that amplified a universal segment of DNA in all species, and then a series of species-specific primers at different points along the gene that would, if that DNA for one of these species was there, these primers would sit there and amplify a specific size fragment. And that's in utility, in actual use, this is what you get. So this is a DNA ladder, or a ruler to determine your size of your DNA fragments. These fragments up here are your control bands, as we mentioned, the universal primers that work on both sides out here and should work on every animal. This serves to say that you had good quality DNA. It would have worked if the DNA from one of these species was present. And we get a different size fragment for a number of these different species. Some of these are not as specific as you'd like. This one product produces an amplification for dusky and Galapagos sharks as well as the oceanic whitetip. But you can identify the two species of Isurus. We can get the poor beagle and a couple other common sharks. And similarly, this can be applied to other taxa. This is some of Deborah's work looking at the hammerhead species. Again, you've got your target, your control fragment that should amplify in all samples if there's good DNA there, and then your species-specific pieces that amplify up in the presence of those animals. So these are actually DNA from other Spherina species, not Mocara, Luini, or Zygena, and then these are other sharks. One thing to notice is the control fragment size is different in the Spherina species than in the target ones, and I'll give you a little hint in advance of the workshop. We have a kind of using these gels to identify species, so keep that in mind. That might help you out with a few things. And then Other people have used similar types of work different ways. Some of the work that we did coming out of the Ecuador workshop was kind of a dichotomous key. So in this sense, we had major groups of sharks, essentially Carcharodontidae, Isurus, Alopius, and Sphaerina. We could, if we had a piece of meat, we could go through, or just a little piece of fin, we could identify what group these animals were, and then go forth and pick the group-specific PCR method to look at hammerheads or thresher sharks. So here's an example of the thresher shark things. Jimmy had a picture of this earlier. We get different size fragments for the 3 different species of Alopius. No amplification in these other species. It tells us that it's working pretty well. There's no control fragments in here, so the one downside is that if you had bad quality DNA, you wouldn't know it. You didn't get an amplification. But if you use the dichotomous key here, you'd see that worked in an earlier thing. You said your species of Isurus had worked earlier, and they didn't amplify in this, and we weren't expecting them to. So I want to reiterate that there's the infrastructure for doing this type of work is pretty minimal. So this is an example of some work that I did in Hawaii years ago looking at identifying billfish eggs, so marlin and swordfish. This is the pre-PCR, this is the pre-PCR side of the lab, the post-PCR side of the lab. The whole lab sits on a tabletop. It's not a huge amount of equipment. You can do it at sea. You can do it in the back of your car if you had the power. The assays are cheap, and again, the results are usually just in a few hours. So just to briefly go over some of the basic protocols, it's as simple as taking a piece of fin, cutting off a small section, placing that into the tube, and then closing the tube. You can see the little tiny, we've got the Helix beads here in the bottom and the little tiny piece of fin. And we plop it into a PCR machine, which basically heats things to a certain temperature. It's an automated oven. We place it in the machine, it cooks it, and then boils a sample to help rupture the cells and get the DNA out. And while we're waiting for that, we can make our PCR reaction, and it's fairly simple. You've got just a few tubes, you've got a master mix, some primers, water. You mix them together. If you can follow a recipe to make yourself a margarita or whatever you want, it's pretty straightforward. Once you have your mix, you put it into individual tubes, and then you just add your DNA to those tubes. Put it back in your PCR machine, hit go, let it run, and you're pretty much mostly there. So the way that we are able to separate the DNA size fragments is we use agarose gels or polyacrylamide gels. These are essentially Jell-O. If you've ever made Jell-O, you heat up something and it forms a gelatinous matrix. The basic way it works is that the DNA of different sizes goes through the gel at different rates. That we use electricity to push the DNA through. DNA has a negative charge, and so we're able to use this to our advantage. And we get smaller pieces of DNA going through faster, larger pieces of DNA go through slower, and if we run this for long enough, we can get a good separation of all of our products. And we usually use, as I said, a DNA ladder or a set of reference standards so that we can compare and see if one of the species of interest is there. So that's basically the overview of genetics. I want to take a moment to say that my lab really wants to collaborate and help people. So this is a quick overview of things. If you want to learn more, I'm making the offer. If you can come to San Diego, make it up there. I'm happy to train you on not just shark ID techniques, but a lot of other basic lab techniques. We have a fairly advanced lab. We've got a couple DNA sequencers. We've got robotics. We've got a whole lot of different things and quite a lot of expertise. I'm also happy to offer advice if you're interested in starting up programs, want to know what equipment you need. It can seem daunting, but we have the information. I'm going to provide this electronically, some of the basic things that you could go out and buy kits to get ready and start doing this tomorrow. We have a number of the papers provided on the memory sticks that everyone's going to be getting to. And so with that, I'll end and I'll take any questions.